This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1187, recorded on January 17, 2025. I'm Vincent Racaniello. You're listening to the podcast all about viruses. Today, it is positively balmy. It's like Miami here in New York. It's 5 degrees Celsius. It's actually above freezing. Also joining me from Madison, New Jersey, Brianne Barker.
Oh, hi. It's great to be here. And compared to Vincent, it's so very chilly because it's 4 degrees Celsius here. But
we're going to go into the negative next week.
I know. Compared to the next couple of days, that's lovely.
Yeah, it actually feels warm today. Oh, my gosh. Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 37 Fahrenheit, which is 2.8 C, and it feels like 23 Fahrenheit, which means it feels like it's -5 C.
We actually get a dusting of snow. Did you get some snow there?
We did. It sort of looked like the ground had a sugar coating on it.
Yeah, you can't even measure it, but it was pretty. So joining us from Montreal, Canada, Angela Mingarelli.
Hello, everyone. Here it's the coldest. It's -4 C, so I think that's like 28 Fahrenheit. And yes, by next Tuesday, it'll be -24 C. So that's like what, -10 or 15 Fahrenheit?
Minus too many.
Minus way too many. Anything negative in Fahrenheit is horrific.
Is that about as cold as it gets up there, or does it get even colder?
Oh, no, it can get to like -40 C, which is almost the same in Fahrenheit, actually, I think. Mm-hmm. I think -40.
Then what happens? Life comes to a standstill?
Then we go out. Actually, below -25, I've said this a lot. Below -25, -30, it's like all the same-ish. When you breathe, it hurts. You have to cover your mouth or else you just cough instantly. Your lungs like refuse the cold air. But it never really goes... Even at -25 next Wednesday with the wind chill, that's the worst part. It'll be -25, but when the wind blows, it's like 10 degrees colder. So your eyes literally just like start watering instantly, and you can't be outside for too long.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here, and we have similarly tropical weather to the other Northeasterners here. It's 38 Fahrenheit, 3 Celsius. So yeah, for once it's not in the negatives and blowing a full gale, but that'll change.
Tropical weather, yes. If you like these programs, we'd love to have your support to keep them going. Go over to microbe.tv/contribute. Our fundraising time is officially over, but you could help us throughout the year. It doesn't have to be just now. Of course, over at Parasites Without Borders, they go to the end of the month, and they're doing a fundraiser for Microbe TV. So if you want to still donate, you can go to parasiteswithoutborders.com. They will actually match your donation, which is cool. Release the viral immunologist in you, the Rosenfeld Laboratory. In the division of viral products at CBER, FDA is looking for a postdoc staff scientist to continue the lab's research, understanding the immune responses elicited during enterovirus infection. If you are an immunologist, microbiologist, or virologist wanting to learn more viral immunology and vaccinology, email Amy at amy.rosenfeld@fda.hhs.gov for more information. And speaking of Montreal, what's up with ASV?
I'm about to sneeze. Somebody else could do it.
Can do that.
Somebody else should just go ahead.
I could just read this. ASV 2025 is going to be held in Montreal at the Palais des Congrès de Montréal, July 14th to the 17th, 2025. As Angela can attest, is a beautiful city. The website is asv.org/ASV 2025. Abstracts are due February 3rd, so I'm not sure exactly when this episode is going to drop, but if you haven't gotten your abstract in and it's not February 3rd yet, get on it. Travel award applications are due February 5th, so better get to that too. And to renew your membership or join ASV, just use your favorite search engine and search for ASV membership.
Angela, is the Palais des Congrès far from McGill?
No, it's very near. It's near the. You guys can come see my lab. I mean, Vincent's already seen it, but anybody else that's here, please come see my lab. And July is very warm. Don't worry. No snow boots, snow pants needed. So everybody, please come.
I'm looking forward to some warm weather.
Yes, it will be.
Of course, I will enjoy it for a few days, and then I'll start saying it's too hot. Right.
We have lots of air conditioning.
I just dream of sitting outside in a warm climate, not necessarily doing it, but just absorbing. I don't know. Can you absorb heat? I guess so.
Yeah.
Sure.
All right.
On cold days, it sure feels like it.
Yeah.
I went to Mexico over Christmas, and it was wonderful. So I am selfishly recently was in a warm climate and then came back here. I was at, I think it was like 29 degrees C when I left Cancun, and then I got to Montreal and it was minus 22. My body was like, for 3 days, my body didn't know what to do. But we're acclimatized now again.
So, Kathy, you able to talk about this new blood test?
Oh, yes. Now I can talk. But let me find where we are in the notes. There we are. Okay. Yeah, so this came across in the newsfeed, and so the link that I have reads a little bit like a press release. But basically, they've developed a blood test for head and neck cancer that's based on detecting HPV DNA from the 6 major human papillomaviruses that are responsible for head and neck cancer. And it's based on measuring cell-free DNA in a blood sample. They use drop digital PCR, so pretty high-tech multiplex probes, and so they can get really good quantitation. And the results come in 5 to 12 days, which at first you think, wow, that's a long time. But when you're looking at, diagnosing cancer, that kind of time is maybe okay. And so then I wrote to the scientist who was mentioned in the press release, Chad Brenner, who I know, and said, can you detect circulating tumor DNA from other HPV tumors using this? And he said, yes, we can. And we're working to set up clinical trials. And so it's still early, but it's promising that it could work for other cancers as well.
So you could pick up cervical cancer this way?
That's what it says. Yeah.
Wow, that'd be
It's
cool.
exciting.
Yes.
Yeah.
Yes.
You don't have to do any swabs.
Right.
Interesting. So what would the idea be? You have like a yearly check for something like this?
I don't know. If you're getting a blood draw for something else, and yeah, maybe.
This is actually part of a family of tests people have been trying to develop for a while, and they're now finally having some success, called liquid biopsies, where the idea is you would look for, there must be some biomarkers coming off tumors and going into the bloodstream and or other bodily fluids, but blood is the one you'd obviously want to sample. And so I think what the ultimate goal is you'd get an annual or semi-annual or whatever blood test that would test for this, 10 different types of cancer.
I guess the question is how much? Sorry, how much is it? And is it actually feasible to add that?
Well, yeah. I mean, those are all questions still to be, yes.
I mean, for years they have had kind of not really good correlates. So the carcinoembryonic antigen, right, for colon cancer. And then I forgot the name for prostate cancer. What's that one? Does anybody remember?
PSA?
Yeah.
PSA, prostate-specific antigen, right? And they're...
Yeah.
So I had my PSA over the course of like 2 years was going up and up and up. My doc showed me, he said, Vincent, this is a problem. We have to have a biopsy, okay? So I had a biopsy. There was no tumor, and then it suddenly went down. So what is it, like a psychosomatic thing?
You just had to threaten it.
For the PSA, I had to threaten it, and it went down? So I don't know. I don't get it.
But isn't there a lot of, I mean, it's weird that it went down because like prostatitis is like common in older men, right? So, but maybe you had like an inflammatory, a low grade,
it wasn't
something inflammatory.
It's just hyperplasia, right?
Yeah.
As you age,
but
it
it's
gets
weird that it
bigger.
would go down if the antigen
Yeah,
is still
but the guy
like,
showed me the next couple of points. I actually went to see a real urologist instead of a GP, right? And he said, yeah, it's going back down again. It's like, what the hell? And in fact, it got smaller, because they can palpate it. Oh my God! So we need better tests. That's the point. The HPV makes sense because it's a virus and viral DNA is floating around, right? But
well, and we now have the colon cancer screening can be done instead of getting scoped. You can just poop in a bucket.
So what do they look for?
They're looking for I think it's a DNA-based test. I think it's a PCR test.
It's PCR as well.
Yeah,
and some antigen, some associated with
they're
cancer,
they're detecting tumor cells that are coming out in the feces that would be that, and doing it sensitively enough that you can now do that. In fact, it's the standard of care for colon cancer screening in basically every industrialized country except ours.
Yeah,
but you can get it in ours.
You can get it in ours.
So what is it?
Yes,
what is it looking for? Do you know what the?
I
DNA
don't know
is?
what the marker is. Cologuard is the name of it.
Yeah, I was going to say that TV commercials don't get into that level of detail.
No, they don't.
Well, there's actually two things, and one is something that you do every 3 years. I have the feeling that's the Cologuard, and then there's something that you do every year, and the one that you do every year is the one that my doctor recommended, and I actually thought it had more to do with blood in the stool sample than anything.
Right.
Right.
There's one that's a looking for blood, and there's one that's doing PCR.
Yeah, and my doctor was not impressed by the PCR one.
Okay.
Okay, Alan, you have something about tecovirimat.
Yes. So, in a surprise to everybody, Japanese regulatory authorities approved tecovirimat for mpox. And this is a surprise because it had been shown in safety studies it was safe, but then people just recently wrapped up a couple of fairly large efficacy studies which showed pretty clearly that it's not doing anything. People got treated. I think they treated people in men who have sex with men in one study. It might have been both studies. Anyway, these are hundreds of people who developed mpox and were, randomized and tested with this drug, and it didn't make a darn bit of difference. And then the Japanese regulators turned around just in January, a few days ago, January 2nd, and announced they're approving it for mpox. And nobody knows why, and the Japanese regulators say they don't comment on specific medicines, so as a nice news article in Science.
Well, maybe.
Up on that.
I don't know why, but hopefully they'll get more mileage out of it.
Yeah. And I mean, this is also something that's been stockpiled for smallpox antiviral, and nobody actually knows if it'll work in that. Animal data for, pox viruses look good, but animal data for mpox looked good too, and doesn't seem to work in humans.
Yeah, it's we're depending on that in case of a smallpox outbreak. Yeah. Very interesting. Okay, on to the literature. We have a snippet for you first, and Kathy's going to give us an overview.
Sure. The paper is entitled MFSD6 is an entry receptor for respiratory enterovirus D68. And so it reports a host molecule that may serve as a receptor for this important virus for which we don't know the receptor, enterovirus D68. The authors identify this molecule, MFSD6, and then show that it's important for infection and that it binds to virus particles. And when it's expressed in non-susceptible cells, it makes them capable of replicating the virus. However, as we'll discuss, the study may lack some rigorous tests that receptor candidates have to pass. And the reason this is important, this whole paper is important, is that understanding the receptor can help with future treatments.
And the paper has, it is not open access, unfortunately. It has 5 co-first authors, Zhizhe Liu, Huili Li, Zhaozhu Li, Delong Gao, and Junfeng Zhao. Senior author is Zhengli Yu, and the team is variously from Jilin University and Yanbian University in Jilin and Zhejiang University School of Medicine in Hangzhou, China.
All right, so this is all about enterovirus D68, which for the last years of my laboratory, we worked on. Amy Rosenfeld, when she came to the lab, wanted to work on this, and so she did, because it was getting increasingly more difficult to work on polio. In fact, NIH said at one point, we're not funding any more polio research. Good, very good. So enterovirus 68 was discovered in 1960-something in California as a cause of respiratory disease. The authors say as a distinctive type of human enterovirus. It spreads through respiratory transmission. But I should remind the authors that rhinoviruses are also classified as enteroviruses. So there are a lot of enteroviruses that are transmitted. There's also some Coxsackie viruses are transmitted by the respiratory route. So read the literature before you write your paper, folks. Come on.
And the enteroviruses, for those just joining us, that's a large group of single-stranded RNA unenveloped viruses.
I sound like the guy from the movie Gran Torino. What was the Clint Eastwood?
Yeah.
Get off my lawn.
Get off my lawn.
Read your literature.
Yes.
So it's a respiratory pathogen, and since its discovery from its discovery in the 60s till about 2014, very few cases found in the U.S. And then in 2014, big outbreaks every 2 years, and a small number of kids got paralysis, which was called acute flaccid myelitis. And interestingly, so that went away during COVID. No more EV68 associated AFM. You don't see it anymore. I think it's population immunity thing. There's no evidence that the virus has changed. In fact, I have to tell you something. People said, when it started causing AFM, the virus mutated, so it starts causing AFM. Really? So you take viruses from 1960 in the present, they are all neurotropic in some way, however you assay this. So there's no acquisition of neurotropism, but it's the easy thing to say.
We just finally got enough infections to actually see it.
Yeah, now they're gone, and I think that it's just a lot of also, there's a lot of cross-reactive enterovirus immunity, and that's what the Rosenfeld lab works on, as you can see in the ad for the position. So I think that may have had something to do with it.
So Vincent,
anyway, yeah,
it mainly affects children, not as much in adults.
Well, it's because it mainly affects children because they're immunologically naive, right? Adults have presumably been infected with lots of enteros that give them cross-protective immunity, right?
But I mean, when children, I guess maybe during COVID, they weren't going to school physically with 30 other kids in a class, so it could just be contact.
Well,
is it oral fecal? Like, how was it transmitted? I forget.
No, it's respiratory transmission.
It's only respiratory.
It's not in the blood. It's not in the feces. No.
Vincent, is this the one that sort of peaked every other year? Or is that
every 2 years?
Every
Yes,
2
that's
years.
exactly right. Go to the CDC chart, you can see. And then during COVID, it stopped. And then after COVID, it did not resume. There are little bits of 68 infections, but no AFM associated with them. And no one knows why. Anyway, so that 2014 and subsequent AFM outbreaks got people interested. A lot of people started working on it. And so part of this is to identify the cell receptor, the cell protein that helps the virus get into cells. And a number of cell molecules have been described as receptors, including sialic acid, ICAM5, heparan sulfate. And the authors say, however, other receptors still exist. I don't know why they say that. How do you know? I mean,
well, they do now, as they're writing the introduction after the.
Why would you say other? I would say it is possible that other receptors exist. So we're going to try. And they say it's the exact receptor that EV68 uses to infect target cells is under investigation. So what are the target cells? Some kind of lung cell, I guess, right? Not HeLa cells in the laboratory. That's not a target cell. So here they try and find another receptor, and they do find it.
Well, in theory, it's also could be some cell in the nervous system if it's neurotropic.
Yes.
I mean, there could be different receptors. Yeah, for sure. We are spoiled by poliovirus where the receptor in the gut and the receptor in the CNS, it's exactly the same molecule, right? But nothing has to be this. Everything doesn't have to be the same, obviously. All right, so they do an interesting screen for identification of host proteins that are important for virus reproduction. Let's put it that way. They take green fluorescent protein and they insert the cleavage site for the viral protease 3C. Okay, so if you put this GFP, this modified GFP into cells, and then infect them with EV68, the fluorescence is quenched because GFP is cut, right? And it can't fluoresce when it's cut because they put the cleavage site in the right place. And so then they introduce a library of RNAi, which targets all cellular genes, and then they infect cells, and they see which RNAis knock down whatever cell protein it is, so that EV68 infection can no longer quench green fluorescence. All right. So they do this three rounds, and they find a protein called MFSD6. Just rolls off the tongue, right?
Yeah.
Of course.
So we called ours PVR, poliovirus receptor.
Well, they didn't get to name this. This is this is an already named membrane protein major facilitator superfamily domain containing protein 6.
See,
that's the advantage of working 30 years ago.
Yes.
Most proteins weren't known and named. So
yeah, that's what I was just going to say. This is the problem with the Human Genome Project having happened.
Yes.
Yeah, so this thing has been named by a description. We don't actually know what it does.
So they use then they use CRISPR to knock out this gene, right, in HeLa cells, and they look at all kinds of things: cytopathic effect, synthesis of viral protein, synthesis of viral RNA, extracellular secretion of RNA, and then finally infectious virus production, which, in my opinion, is really all that matters. And you know, the infectious virus production isn't zero when you knock out this receptor. And they don't comment on this at all. And it could be that this is the virus that was used to infect, in other words, residual. Or it could be that there are other receptors, but you can't tell because they don't give you enough data on that. So I'd say this is
Or it could be, at least in the siRNA experiments, that they didn't get a very good knockdown and there's some residual protein. And that should be really easy one to show.
Well, this is CRISPR now, right?
Right, but the earlier one times as well.
Right.
So the CRISPR knockout should be zero unless there are some of the issues that I just talked about. Anyway, then they can put the gene back into the MFSG6 gene back into these knockout cells, and it restores normal reproduction. But we're talking a couple of logs of virus, between 4 and 5 logs in the knockdown cells. So it's not insignificant. And they do not comment at all on that, which is weird because the reviewers should have asked for it. What do you think is going on here? All right, so those are HeLa cells, which irrelevant to any virus.
Irrelevant for ultimately, but a great place to start because everybody's got them on hand and they're easy to culture and manipulate.
I tell you, I am guilty. I spent most of my career studying poliovirus and HeLa cells. Right?
Sure.
I was trained as a postdoc because you could grow them really well and you could do all kinds of biochemistry. Then I get to Columbia, and after a number of years, I start to study the interferon response to polio. I write a grant, and they say, "You're out of your mind. You can't use HeLa cells to study this. It's irrelevant." And I said, "Oh, yeah, you're absolutely right. You're right." Because I'm thinking like a biochemist, all these years. Anyway, they try some other cells, respiratory cells. These A549s, which are lung cancer cells, not the best. But then they go to normal immortalized respiratory epithelial cell lines, bronchial epithelial cells, and some others, and they find that knocking down MFSD6 knocks down the amount of virus you produce, but doesn't make it zero. in any case. They can show that if you add virus to cells, you can show that it binds to the protein by immunoprecipitation or immunofluorescence strategies. So you add the virus to cells and it will bind to it. So that's good. That's what a receptor should be doing. They look at virus entry into cells, and it's also decreased in the knockout cells. Now they use these cell lines, Viro cells, vervet monkey cells, right, Brianne?
That is correct. Yes.
What's the other grivet?
Isn't it the grivet?
Is it the grivet? So they're grivets and vervets, right?
Yeah,
two different kinds of.
They are.
These are Old World monkeys or New World? I forgot.
Old World. They're basically, there used to be sort of the African green monkey group, which we have now realized is actually many different species. But
Viro is made from one of those two, and there's not another cell line made from the other, is there?
The problem was there was a species, and I'll just say hi, Jens. There was a species that has since been broken up into two others. Yeah. And the one that I usually think more about is COS cells that we only know is from that parental. We don't actually know which of the two real subspecies the COS cell is from. I believe Viros are specifically grivet, but I haven't been reading that literature recently.
Anyway, so Viros apparently don't make a lot of virus when you infect them, but if you put in MFSD6 gene, it increases it. But it's not zero to begin with. There's a couple of logs already. And so again, is that input virus? Is it some other receptor? We don't know. They don't address it. The same thing with MDCK cells, which are canine kidney cells. They do make some logs of virus, and it goes up when you put MFSD6 into it, and I don't know why that is. So this is an interesting protein. It has 12 potential transmembrane domains. So it's a multi-pass plasma membrane protein. So they do some structural prediction, and they find an extracellular domain that they think would bind virus. And so they take that sequence and make a fusion to the FC portion of an antibody. Right? So now you have this MFSD6 fused to the FC, and that will bind to virus particles.
So I don't think it's the whole FC. Yeah. It's just part of the FC, one of the heavy chain constant region domains, the third constant region domain.
Well, that's another construct they make. First, they just use FC.
Okay.
Right. So the way I would do it is like, here's your antibody, and the part that's my wrist down to my elbow is the part where we're talking about. It's the FC portion of it. And then, as Brianna is saying, later they're going to use just the part that's like the very end of my forearm toward the elbow. And then instead of having the antibody combining region, they put on this domain instead. So it kind of looks, if you draw a structure of it, a cartoon of it, it looks like an antibody, but it's not because it's got different parts.
Yes, I wish I had my little stuffed antibody.
Yeah, I had mine back there, but I had decided I would just do it with hand and elbows.
Where did your antibodies go, Brianne?
It's in a drawer that I can't reach easily right now.
They decline over time.
They do decline. That was a title. That was the title of a recent immune. Where do the SARS-CoV-2 antibodies go? So then they make this statement. We upgraded and redesigned, upgraded this protein. So they added now the constant region 3.
Bug fixes and usability improvements.
So you have FC with constant region, heavy chain 3, and they say that you get better dimerization and blah, blah, blah. And they do the same. This does the same thing. It binds virus. It blocks infection of a variety of different cells. So it's clearly binding virus, and it is interfering with virus infection. Okay. And then the last thing they do is to see whether this works in a mouse model of an infection. So they have a lethal model, and I have a little trouble with this. Okay. They infect the mice intraperitoneally. Okay. I mean, it's a respiratory virus. Okay.
Why wouldn't they do intranasal?
Yeah, exactly. I don't know why. They put virus intraperitoneally and it kills them.
IP is easy for a biochemist.
Intranasal, yeah, I guess. But actually, no, because you have to scruff the mouse and hold them. That intranasal, they're sleeping already. I don't know.
Yeah, but that means you have to do the anesthesia.
They sometimes will really like sneezing.
It's true, they sneeze a lot. But then it's not relevant though, like Vincent says, intraperitoneally.
I don't think there's any relevance to this. I mean, first of all, we don't know why they're dying. Maybe they're dying for maybe it's infecting something else. Who knows? Maybe it's getting into the CNS, right? But they all die within 10 days or so if you put the virus IP. And all they do is look at survival. They don't look at virus reproduction. And then they inject this FC fusion with the MFSD6 IP also, and it protects the mice. Only a few mice die. Okay, so I don't know what that means, right? So we know this thing blocks infection. So yeah, you're putting it in. So basically, the mouse is like a cell culture. Where you do the same experiment, but you have no idea what the physiological or pathogenic relevance of that is, So,
so at best, it could improve disease outcome, improve survival rates, maybe. But there's obviously some other receptor that not only MSD? I can't even say this. MFSD6.
MFSD6.
Yeah. So maybe it's involved in viral entry, obviously, but not the only thing.
Yeah,
I think that there probably are others, but they don't really talk about what they might be doing. I mean, first of all, if this thing causes AFM, acute flaccid myelitis, the virus is clearly getting into the CNS. And the work Amy did in my lab years ago showed that it infects multiple neurons and astrocytes in the brain of mice. So what's the receptor there? They don't actually address that. They could do IC inoculation. Or best would be to put it respiratory and have some of it go to the brain and cause paralysis, but that's not happening here.
It might also have been nice to see some tissue staining.
Yes,
exactly.
I agree.
Maybe even,
system.
Some of these folks are at medical schools, they could have gotten lung biopsies from human and seen if this thing is actually there. But overall, yeah, I think I'm pretty convinced that MFSD6 is something that the virus seems to like having.
It binds to, right?
It definitely does better when that's on cells in these systems, but I'm not sold that this is a major or certainly the only receptor. There's obviously more going on here.
And so when we identified the poliovirus receptor, which was 1990 or something, Kathy Mendelson and others, the last figure in the paper was a northern blot of human tissues where the probe was poliovirus receptor DNA, just to see if it's produced in human tissues, right? And it is. Unfortunately, it was in all the tissues we looked at, but it was in spinal cord and brain and intestine and so forth. Yeah. And that's absent here. We don't have any human tissues that are examined. They actually look at, this is the modern way of doing it. They look at transcription databases now, right? Where people do RNA-seq of various tissues. And what do they say here? It's multiple databases indicate that MFSD6 mRNA is significantly expressed in a variety of tissues, including lung, GI tract, blood, and nervous system. So I guess it's just a different assay from the northern blot, right?
Yeah, I will admit that, especially with the sort of mouse experiments, their use of the FC fusion really kind of lost me. So they basically just made this domain of interest of the protein, and put it on an FC portion. Basically got a dimerized version of it. And in some ways, it felt like they made the FC portion so that they could purify that protein.
Yeah, maybe.
It felt like they got it sort of to use as bait so they could purify out this domain that they've made. Because they don't show what happens if you use a non-FC binding, just the actual protein. They do a bunch of work to use just the CH3 in order to get around things like FC receptor binding and to make it small enough that it can penetrate into different areas. But they never actually just make the protein, which I guess makes sense if it has 12 transmembrane domains, and look at what the protein does. Is this small domain usually a dimer, or is it ever found as a dimer? Here, it's always acting as a dimer. It just, I was sort of, again, thinking that this seemed like a very biochem focused way of looking at some of this without thinking about the in vivo biology.
Good point.
There's to talk about there being more than one receptor for this virus. And they say this phenomenon deviates from the conventional one virus, one receptor model. Well, again, can you please read the literature before you write your discussion? Plenty of viruses require 2 receptors. Let's see. Who can identify one? Brianne.
HIV.
Kathy.
Adenovirus.
Yeah. So it's not conventional to just one virus, one receptor.
Yeah, I sometimes in grad school, my advisor would make some comments about people needing to read the literature, and I would stick up for them. I had a phrase that I would use to stick up for them. That phrase is not appropriate in this situation. I feel like my undergrads should be able to pick up a couple of these things.
But, I mean, this should have been picked up in review also.
Yeah.
So this is feeding to my contention that the review process is getting sloppy because there are too many papers now submitted, too many journals, and people don't have time to read it properly. So,
I mean, the flip side of that is it's harder and harder to find reviewers.
Sure.
Everybody's so busy that they don't want to review, and so you find somebody who does review, and then they're in a hurry because they're also just as busy.
Yep.
I was also not thrilled with the degree to which they seem to be enthusiastic about this MFSD6 as an antiviral.
Oh, yes, really.
They're a little too amped up about that. And, when you haven't even figured out what your escape mutants look like, because, if you throw this at the virus, if the virus needs it, it's going to get around it. And you'd like to know how that's going to happen so you can try and address it ahead of time. But none of that.
So that's what we did in our lab. We took the polio receptor. We made a soluble version, right? If you add that to virus, it blocks infection. But you always get a few plaques, and if you pick those, they have mutations in the genome that get around being neutralized by the soluble receptor. Now, you may think, if you're thinking carefully about this, well, how does it infect cells if it's resistant to soluble receptors? Obviously, it's different. The soluble version is different from a cell-bound because it can infect cells. And so I think that you don't need a lot of reduction in affinity to get around neutralization with a soluble receptor. And that's still consistent with infecting a cell membrane-bound version. But it can happen, yeah. All right. So I think, every paper we do isn't perfect, but you can learn from mistakes, right? That's the whole point. You can say, well, this should have been done this way and this should have been done that. We don't often do that, but that was one of those examples. Now, this next paper is really good, I think. And Kathy's going to summarize it for us.
Sure. The title is Anti-herpetic Tau Preserves Neurons via the CGAS Sting TBK1 pathway in Alzheimer's disease. And so this paper strengthens a connection between herpes simplex virus type 1, which I'll say HSV1, and Alzheimer's disease. And in particular, the authors looked for HSV1 proteins, not nucleic acids. So a lot of the evidence in the past has been relating to viral nucleic acids. And so they use this really cool technique that allows them to look at protein-protein interactions in a new way that I don't think we've talked about, decrowding expansion pathology. So we'll tell you about that in a little bit. But so because they can look at these protein-protein interactions, they're going to tie together HSV1 proteins and phosphorylated tau. And tau is one of the protein hallmarks that you've probably heard about with respect to Alzheimer's disease. And so the presence of the virus, the authors go on to show the presence of the virus increases the tau levels and that this suppresses infection. And so why this is important is it was previously thought that tau was only harmful, the presence of tau associated with Alzheimer's disease. But this is suggesting that the presence of tau, phosphorylated tau, can suppress infection. And so maybe it's part of the immune defense. And obviously the overall importance of the paper is that we'd like to know a whole lot more about Alzheimer's disease.
Yeah, the points that Kathy made there about tau being related to the immune system were definitely the things, of course, I got really excited about because they're talking about it, in fact, being involved in a pathway that I work on in my lab. And there was one experiment in this paper that if they hadn't done that, I was going to just run to the lab and do it.
And this paper is open access, and the lead author is Vanessa Hyde. Senior author is Or Shermesh, and they and their collaborators are at Hebrew University of Jerusalem, University of Pittsburgh School of Medicine, Tel Aviv University, and Carnegie Mellon University.
So when I came across this paper, I said, is this the one that Kathy just sent me? Because Kathy had sent a paper also on Alzheimer's. Mm-hmm. And that one is also interesting. We'll probably do that on TWiV. It's all about head injury causing viruses to reactivate in your brain, and that's associated with Alzheimer's. But Alzheimer's pathology is driven by what are called neurofibrillary tangles of protein made of phosphorylated tau, okay, and beta amyloid. And
so the tangles, right. So tau forms tangles and beta amyloid forms plaques.
Right, plaques, tangles and plaques. As the way they write it is, I'm not sure. For neurofibrillary tangles made predominantly of phosphorylated tau and beta amyloid plaques or oligomers made predominantly of alpha amyloid beta protein. But what triggers these, we don't understand, right? And over the years, people have suggested viruses, bacteria, fungi, parasites could all be.
Isn't there a lot of controversy now in Alzheimer's that it's not only these plaques and tau, and that there can be a lot of other things? Because even in animals, like I remember in vet school, and all like animals that we looked at their brains, they all have beta amyloid plaques, and there's no sort of like neurodegeneration whatsoever. And even humans, like older humans, have beta amyloid plaques. So is it like there's something else? It's not just having that equals having Alzheimer's. I think it's important to.
There's a lot of other things going on here. Mm-hmm. So people have mentioned the neurofibrillary tangles with tau, and the idea there is that tau is basically polymerizing. So it's not as though just having tau is necessarily a problem. It's having it polymerize in this problematic way. Sometimes seems to be associated with Alzheimer's disease. Same thing with amyloid beta. And yes, there are definitely a lot of places where this is, there's sort of some controversy about this. But at least in terms of sort of the epi, there's good linkage. One of the biggest problems is that neither of these things are things that you can detect in a living brain. And so they're all things that are detected only post-mortem and then tied back to, the patient was having X, Y, or Z symptom.
Yeah, well, I'm not surprised, but this paper focuses on tau and beta amyloid as key processes, right? But
now, most people who work in the Alzheimer's disease field spend their time focusing on amyloid beta and tau, and often in cell culture models like the ones that are shown here.
And when tau forms these tangles, it's heavily phosphorylated.
So viral infections have come up with the strongest correlation, particularly encephalitis, like herpes simplex encephalitis. So herpes simplex encephalitis is a rare event, but it can happen. And survivors of encephalitis have an increased rate of development of Alzheimer's. And there was a big study done in Taiwan a number of years ago, which we covered on a TWiV episode. So they looked at medical records and found that HSV infection without any treatment increased the risk of dementia by 2.56-fold. And when treated with anti-herpetic medications, that additional risk disappeared. So now there are actually a number of clinical trials in progress, seeing if you take people who are in early stages of AD development, if you treat them with acyclovir, does it make a difference? One of them is being done at Columbia, I know. So, Alzheimer's pathologies are linked to the presence of herpes simplex virus DNA. Infection does induce alpha amyloid beta production, and it induces phosphorylated tau, and these are in cell culture models.
And both of those things make me start to think, is that because these amyloid beta and phosphorylated tau actually have an have some immune property? Is the immune system trying to make those because they're useful?
Yes. Yeah, so one of the ideas was that these are immune responses to infection, which somehow then lead to a very bad disease, right?
Don't HSV-1 infections, isn't it about half of us have HSV-1? It's just in some sort of low-level latent form, or am I wrong?
About over 80% of people are seropositive, right?
They are, okay.
And have neurons, ganglia. So peripheral ganglia contain viral DNA, not the
active
CNS.
Infection.
I mean, the incidence of herpes encephalitis is about 1 in 200,000 in the U.S. because basically you have latent viruses in the trigeminal ganglia. When they get reactivated, they go to your lips and nose and they make fever sores. But sometimes they go in the wrong direction. They go into your brain, and then you get herpes encephalitis. And that's where you get the increased risk for Alzheimer's. And years ago, we did a paper from Casanova at Rockefeller showing people who get herpes encephalitis, they have mutations in the TLR7 pathway, which failed to respond to herpes infection, and you get higher viral loads and more likely to get into the CNS. So the thing is here that they say it's a challenge is that you never see infectious viruses in brain samples, right? So what's going on?
And just looking for the DNA in brain samples is not very informative because, as Vincent said, 80% of us are walking around with this, the viral DNA is just sitting there. It's never going to do anything.
In fact, if they do studies of Alzheimer's versus non-Alzheimer's patients, you'll find herpes DNA in both. So that's not very useful, right? So they say we should be looking at proteins instead, and we should look at it in a way where we can see where the proteins are, not grind up brain and do a PCR for viral DNA. And that's where this decrowding expansion pathology, DexPath, comes in. Now, Alan, you had a nice description of this in your
so this is, I looked this up and I said, wow, this is cool. It was just developed about a year ago, or just reported about a year ago. It basically, it physically expands cells. It's a chemical treatment that you do on say, a tissue sample or something. And it physically expands the cells so that now you can probe protein location, and in blowing up the cells, it keeps their structure and their relative positions of everything intact. So it's
how is that possible if you're expanding the cell?
That you'd have to read the original technique paper.
It's a subset of a technology that was previously known, and someone in our department has been doing it, so we've heard about it for a couple of years. Expansion microscopy.
Yeah,
and in there, the proteins get digested and and this, the proteins don't get digested; they get somehow softened.
Yes.
And so, it's a really cool way. And the person in our department who's done it has had to do a lot of controls to show that it's not creating artifacts and things like that. But you can do, like, if he does the expansion microscopy too many fold, then it starts to wonk out. But and so this is a variation on that.
So really cool. They can embiggen the cells, and use achievable magnifications under the microscope and conventional probes and track where the proteins are in individual cells in, say, a brain section, which is what they're going to do.
Okay. So they start off by showing you, if you take 3 Alzheimer's, advanced Alzheimer's, and 3 non-Alzheimer's patients, brain samples from them, you can find herpes DNA in all of them. So that's not very useful. Then they say, can we detect herpes viral proteins using Western blots, again, on brain samples. And they looked at a variety of herpes viral proteins, including transcriptional regulators and glycoproteins. And they find them all. They find them all in these brain samples. And they end up focusing on ICP27, which didn't have a significant difference between no Alzheimer's and advanced. You would think that would be not, but there's a reason why they focused on that later. So then they use this expansion microscopy to, first they use conventional immunohistochemistry to look at neurons and phosphorylated tau. This is in the hippocampus. And they don't see a difference in phosphorylated tau based on, again, normal and Alzheimer's. They both have phosphorylated tau, as was mentioned earlier. And there's no difference. If you have severe disease or not, you still have phosphorylated tau. And they say these challenges may also help explain previously difficulty in detecting proteins in human brain samples. So they use DX path now to
right. So they're going to expand the minds.
They're going to expand their minds to look at these proteins. And
I just figured out in these figures where they have there's a little blip that I couldn't see until now, and then it goes into it's a little brain going to a bigger brain, showing you that it's expansion.
Oh, the expansion.
Yeah. It's this white thing in the bottom right.
So now when they look for phosphorylated tau using this expansion, they see decreased signal in the no Alzheimer's controls, which is good because that's the way it most likely should be if you think p-tau is important, right? And in advanced Alzheimer's, they see a big increase in phosphorylated tau expression. So they think that this DX path is just the thing to look at this. They think that the tau. So their opinion is that the phosphorylated tau signal in normal tissues is false positive, right? Because of this result. Now, whether you believe that or not is a different story, but that's what they think. So then they look at these herpes viral proteins using this technique across different brain regions and disease severity. They mostly look at the hippocampus and entorhinal cortex because there's a lot of Alzheimer's in those tissues, and they quantify the protein by counting positive pixels. Okay, and they find that ICP27 signal increases with increased Alzheimer's severity. So between mild and advanced Alzheimer's, it doubles. The number of pixels doubles. So that's why they focus on the other proteins. The other viral proteins are not so impressive. So that's why they end up focusing on ICP27, which is a transcriptional regulator of the virus, by the way. All right. So then they want to know what cell type is making ICP27, and they focused on neurons and microglia. All right, so neurons are where the viruses multiply, and microglia maybe can pick up particles and you would see the proteins in there. And they find high colocalization between ICP27 and neurons in all stages of Alzheimer's disease, and less so in no Alzheimer's disease. Right, it's 58% decrease. And they also find low colocalization between ICP27 and microglia, and an increase in advanced Alzheimer's. And they think, again, this could be because these microglia are picking up the HIV particles, or maybe they're being infected. Who knows? Who knows?
Yeah, where do they think, I don't know if they said it, where they think the ICP27 is coming from? Actually, the cell type. They didn't say, right? They don't know?
Well, it would have to be neurons, right? Mm. Neurons are infected by
It
simplex.
should be the neurons.
So I presume that they are,
okay, not the microglia.
They are making it. You know, I always thought that if you had simplex replication in the brain, you're in trouble. But what they say here, there's a word they use which I may have skipped over. You know, it's basically a subclinical infection, but they call it something else, which I thought was pretty funny. Anybody remember what that is? Maybe I'll come across it later.
No, I was just Googling whether or not microglia could be infected by HSV1, and it looks like maybe it looks like there's some abortive infection.
Yeah, I think they kind of
infection.
They kind of address that in their discussion. Is it that it's engulfing the virus, or is it being infected by the virus? Which is always a question when you have macrophages.
But you know, if you had an active infection of neurons, you'd get encephalitis. Okay, and you can't have that all the time. So there has to be low-level protein production in the absence of virus particles, because the particles would spread and infect other neurons. So that's what, because when I first heard this years ago, I thought, how can people survive encephalitis? I said, no. So David Knipe said to me, no, they don't have active encephalitis. They have very low level and perhaps periodic reactivation, very low reactivation, which makes these proteins, and that could be what's going on. Anyway, then they say, where's tau? Where's phosphorylated tau? And they say, the samples, the advanced Alzheimer's are positive for, and tau is phosphorylated at many sites, okay? Lots of amino acids, serines and threonines can be phosphorylated. And they say they were positive for all the phosphorylation sites. So they looked at one or two phosphorylation sites in the subsequent. And they end up staining by this technique for phosphorylated tau and ICP27. So in advanced Alzheimer's, they see a good colocalization between ICP27 and phosphorylated tau. And you can see that in the pictures. Same thing in the...
Sorry, Vincent.
Yeah, go ahead.
No, it's just interesting that even no Alzheimer's, if we look at ICP27 and mild Alzheimer's, it's not that much more. Like even with no Alzheimer's, there's still ICP27, which is, so it's not like all or nothing. Like it's there.
No, it's always an increase. You know, it's so I always tell you a significantly increase of 8-fold. So in the entorhinal cortex, the colocalization significantly increased 8-fold from no Alzheimer's to advanced. So it doesn't mean it was 0, right? It just increased 8-fold.
Right. And Vincent's gotten trapped by the language that they use, because it's not that it's increased, it's that the level is higher. It's not like they take one sample and they can show that it's increased.
Yeah.
It's two different samples with and without Alzheimer's or mild and advanced.
Right. Yes, because they're two different people, right? So that you can't say it increased. Same in the hippocampus. They find these. All right, so ICP27 and phosphorylated tau co-localize in Alzheimer's brain samples, and the colocalization goes, what's the word, Kathy? Not increased, but is higher.
It's higher. The levels are higher.
In disease, with the disease severity. The other thing that's interesting, there's no colocalization of amyloid beta with ICP27, which they say is these plaques are a canonical pathology of Alzheimer's, yet we don't see localization with ICP27.
But in the picture they show, there is ICP27 next to the plaques. There's just not as much, unless I'm looking like in figure 3. I'm sorry for like specifying, but
I think that there is some, but it doesn't go up. It doesn't go up in Alzheimer's, right?
Yeah, I guess so. Also, some of the figure legends, not the legends, but the actual figures, the panels, it's a little bit crowded. They should have had like a little bit more space for some of these panels.
It's very crowded.
Crazy,
They could have decrowded their figures.
Yeah, the figures are crowded, and some of the legends are on the next page, and I kept having to flip back and forth. But that's just a product of the immense quantity of data they crammed in here.
It's a lot.
I was lucky because, me being a dinosaur, I printed it out, and the legend was always on the opposite page from the figure, so I didn't have to flip pages. It was great.
That is a treat.
They compare p-tau in neurons and microglia, and what they find is that p-tau is localized to neurons all the time, no matter where you are in Alzheimer's spectrum, right? None or intermediate or advanced. There's just pretty much the same co-localization. But it goes up in microglia with disease progression. So p-tau goes up in microglia with disease progression, which may be, as you're going to see, if this is really an immune response, that's those cells are doing that, right, in part. Okay, so they think, all right, so maybe herpes simplex infection leads to tau phosphorylation. Maybe that's their idea now from what they've seen. So they make neurons from human-induced pluripotent stem cells. And then they infect and they look at tau phosphorylation. Here, you don't have to use any fancy cell expansion thing. You can just do a Western blot and look for phosphorylation. So compared to uninfected neurons, so now they can say increase because they have the same cells, right? Compared to uninfected neurons, the phosphorylated tau goes up 183% after HSV1 infection.
This is cool, yeah.
They say these data suggest that infection induces tau. Yeah, I would say so. Okay. All right. Then they do human induced pluripotent stem cell derived 3D brain organoids, right? Which, as they say correctly, recapitulate some aspects of the 3D structure of the brain. Some people call them mini brains. No, they're not mini brains.
That's questionable.
You don't get the whole thing. No.
Yeah, it's an organoid is not the actual brain.
It's not a mini brain. So they're going to infect these human organoids. 6,000 PFU per organoid. They put with herpes simplex, and then they fix them on days 3 and 5. And again, you get an increase in phosphorylated tau. It's not as much. It's 11% on day 3 and 26% on day 5. But this is it's an organoid. It's not just neurons and microglia. It's other cell types as well, which may not be infected by herpes simplex 1. So then they treat these organoids with antiviral drugs. So we have good antivirals for herpes simplex virus. And as you would expect, the drug treatment leads to an inhibition of the increase in tau phosphorylation. So that's good evidence that the virus infection is doing that. And then they say, well, in the human brain, you maybe get periodic reactivation of latent herpes genomes, right? So the infectious cycle begins and you make protein. So let's see if we can do that. So they infect these organoids. They add antiviral drugs to suppress reproduction, but this puts them in a latent state, the genome's in a latent state, and then they induce them with another drug. And they find that even after this reactivation, they can get phosphorylation of tau. So they conclude that herpes reactivation leads to an increase in tau, but it does not lead to an increase in phosphorylation, interestingly. Okay. Then they switch to rodents because it's really hard to make brain organoids, basically. they say brain organoids have approximately a 4- and 8-fold slower turnaround time. They're really hard to make, but they can use rodent neuronal cultures.
It's much easier to make rats.
Rats.
It is. There are people here who make these cultures to look at tau and amyloid beta. So
now these are 2-dimensional primary cultures from the rat brain. But the findings with tau and herpes and ICP27 align with what they've found in human cells. Okay. All right. So they say, is it possible that tau, once it's turned on, will reduce ICP27 production? So they add tau kinase or phosphatase inhibitors to primary neuronal cultures that were infected with HSV1. And so they say neurons immersed in CDK inhibitors. That's an interesting way to put it.
I mean, that is, I guess, what's happening in that.
Yeah,
well, it just added to the medium, right?
Yeah.
Right.
Yeah. Kabath.
But anyway, immersed before infected show a 96% increase in ICP27 when compared to inhibitor-free. So in other words, if you inhibit phosphorylation of tau, ICP27 goes way up, which implies that phosphorylation of tau is inhibiting ICP27 production, which is kind of interesting, right? So tau kinases are decreasing ICP27. So they say, well, maybe tau inhibits protein expression in general. So let's look at this. And they look at they look at other proteins. No, it inhibits tau phosphorylation. So we just looked at expression before. They looked at what's the difference between decreasing expression. We hypothesized that tau phosphorylation inhibits viral protein expression.
Yeah.
Okay.
So what they're basically
they mutate a phosphorylation site. Right?
The idea here is that tau is somehow doing something to be antiviral.
Yeah.
And so again, this is sort of getting to that hypothesis that tau is useful and in some way we want it. It's antiviral, and as we said at the beginning, perhaps these neurofibrillary tangles are this immune response going awry. And so this is some evidence that says, okay, tau is in fact specifically reducing this viral protein. So tau does seem to be doing something antiviral. Yeah,
they have an amino acid change in tau, which, so the wild type tau reduces expression by 54%. And if they make this one amino acid change, it's reduced even more. I don't know what this change, that says tau P301L. I'm not sure what that is. It's not a phosphorylation site, obviously, but maybe it's already known to affect tau phosphorylation. But if you make that change, you get an increase in the reduction of ICP27. Now, this is interesting next. So what's the role of tau, phosphorylated tau, in the survival of neurons? So they put tau, or this altered tau, into neurons infected with HSV1, and they quantify neuronal death. So control neurons, the death rate is 9.24% after HSV infection. But if you use this altered tau, it goes up to 84%. So the conclusion is that phosphorylation of tau prevents neuronal death induced by HSV infection. So simplex infection is going to kill the neurons, and tau phosphorylation prevents neuronal death.
So it's protective in this case.
It's protective.
Yeah, so
which
again,
is interesting because we associate it with disease, but in a different disease downstream.
Right.
Alzheimer's.
So again,
But
we're it's sort of this idea of, hey, tau is doing something useful in the viral sense, and so perhaps we might hypothesize the immune system actually wanted to make tau.
Yeah.
Perhaps tau is actually the product of some immune response. It's useful. Look at all these things it's doing to restrict a virus.
Maybe it seemed like a good idea at the time.
Yeah.
We live too long. If you don't get Alzheimer's at a young age, right?
Nope.
It's a problem.
Also true.
Evolution expects us to be dead by 40, so this is all.
That's right.
It's true. I'm on my last legs in a few years.
So they want to know whether they can link other immune responses to tau, right? Because this looks like a protective effect.
Yeah, so it looks like your immune system made tau on purpose to do something. So can we figure, or it made phosphorylated tau on purpose to do something. Can we figure out how the immune system made that phosphorylated tau?
Right. And it's known that herpes and other DNA viruses are recognized by cGAMP synthase, which is a sensor for viral DNA in the cytoplasm of the cell. And this activates cGAS sting, which then results in the induction of pro-inflammatory antiviral cytokines, chemokines, interferons, and so forth. We've talked about this a lot.
Yes.
Before.
And I definitely have not been down a rabbit hole of Googling some pieces about this while we've been talking. But as someone who spends a lot of time thinking about cGAS sting, two sort of things that are really important here is that one, cGAS is known to respond to DNA viruses, of which a herpes virus is an example. And so that's one nice link here. But when I draw out the cGAS pathway, which is actually behind me on a whiteboard, one of the things that we always draw is a kinase called TBK1. So cGAS signaling turns on a kinase, TBK1. And that's important to think about because, oh my gosh, the way you'd get a phosphorylated tau or a phosphorylated any protein is that you have to have a kinase. Mm-hmm. And so there is a kinase here. There are actually a couple, but TBK is one that's particularly important here that could be implicated. And it's a well-known kinase, TBK1.
Now, also downstream of TBK1 are NF-kappa B and IRF3, which are essentially transcription factors that get activated and turn on transcription of interferons and so forth. So they image NF-kappa B in these brain explants. They image NF-kappa B and IRF3, and they compare no AD to advanced AD. Alan Dove. Oh, I just realized it's your name, Alan.
Yes, it's it. Yes.
NF-kappa B expression increases 5.4-fold to advanced AD. Not increases, but goes up. And
is higher.
Is higher, and 4.7-fold in the entorhinal cortex. And the same thing with IRF3. It's 5.2-fold higher and 3.6-fold higher. And they co-localize. So NF-kappa B and IRF3 co-localize with ICP27 and phosphorylated tau and advanced Alzheimer's disease. So from this, they say it could be that these innate sensors are involved in tau phosphorylation. So they do an experiment where they take neuronal cultures and they activate. They have a compound that activates the downstream cGAS sting pathway. Okay, so
this is the experiment I would have done if they hadn't done it.
You've got it in your fridge. This stuff. 2', 3' cGAMP sodium salt.
Yep.
Which activates the downstream, and they get increased phosphorylated tau when they add this salt. So they say that links directly the cGAS pathway and tau phosphorylation.
Yeah, and they do a nice job of looking at imaging to look at tau tangles or tau polymerization. They do a nice cGAMP dose curve. Then they add in a TBK inhibitor to show that kinase is important. So I thought that this was done really nicely.
Yeah, they use an inhibitor of TBK1, which is downstream of cGAS, and that phosphorylates IRF3 and NF-kappa B. And treatment with the inhibitor before they add their activator of cGAS sting and eliminates tau phosphorylation increase. And that's their last experiment.
Yeah. And so I think this experiment is fascinating because it's basically showing that at least in these neurons, general activation of this cGAS pathway is leading to tau phosphorylation. And so this says, the tau phosphorylation and in some ways the production of these oligomers is an immune response that's being made in these cells.
So how is tau antiviral?
That's a great question.
Tau is like it's a cytoplasmic microtubule associated protein, which I just looked at. I was like, oh, it's inside the cell, right?
Yeah.
This is like cytoplasmic, as opposed to amyloid beta, which are plaques that are deposited extracellularly. So it's not even, they're not even necessarily the same, but tau in itself, it's associated with microtubules. So like, what does that have to do with the cell being antiviral? Like, I'm trying to think about what that could mean.
No, I don't know. And, similarly, is this just a neuron thing? Do you make phospho-tau in other cells?
Yeah, like in the liver. Could we get phospho-tau? I have no idea.
What makes it so that this phospho-tau doesn't cause problems in everybody?
So it's mainly a neuronal protein. So that explains that.
Right.
But I would like to know how. So you could do this in neuronal cells, right? You can try and understand how tau inhibits viral gene expression.
And I expect they're doing that.
Yeah, they should.
And another question is, okay, so tau is good. Phospho-tau is this defense mechanism, but then how do you get to the point where you're producing these tau tangles that cause Alzheimer's disease? And they hypothesize in the discussion that it may be that repeated activations, if you keep activating the herpes virus, this chronic state of it may lead to the formation of phospho-tau tangles, so it would be collateral damage from the immune response.
In a way, it's another side of the if there's too much of the immune response, it might not always be good.
Yeah.
It just in a different way of thinking about it.
Well, if you're 40 or 30, it's fine, right? Problem is
Well, do we know that? Could another virus be doing the same thing? Are there other viruses? They're looking at HSV1, but potentially other viruses could also be causing phosphorylated tau increases. Sure. We have no idea. And that could also be leading to Alzheimer's, but via another virus or pathogen, TB.
I guess any virus that gets into the brain, right?
But is that true?
Wonder
we don't know.
So, polio virus gets in the brain. I wonder if that induces tau.
I don't know. Yeah.
Well, can't do the experiment because they're stopping polio research.
It's true. Your lab is closed, Vincent. We can't do it.
It's been closed for a while.
No,
but there are other viruses that get in the brain, and you could look at that.
And is this distinct to DNA viruses, or does it happen with RNA viruses too? It's true.
Yeah, well, the RNA viruses are not sensed by cGAS.
cGAS, exactly.
Right.
So maybe you don't get phospho-tau.
Not exactly.
I mean,
there's some that are.
There are some.
So
okay. No, it's the immune system, right? Sorry, no absolutes.
If they damage mitochondria and the DNA comes out, right? Yeah. That can be sensed.
Right.
Yes.
Okay.
Yeah, that's like dengue.
That is exactly.
Something like that.
Exactly.
So that could happen in the brain too. Yeah. Well, it's easy enough to see if tau is phosphorylated in cells in neurons infected with a variety of viruses. Yeah.
Yeah.
Mm-hmm.
So do you think we should just give people TBK1 inhibitors their whole lives?
I think it's a little early to jump in and start giving people something based on this.
The problem is, if you take a person with early Alzheimer's, you already have tangles.
Yes.
And you can't probably reverse those, right?
Not that we know of yet.
I mean, if you dephosphorylated all the tau, would that reverse the tangles? I don't know.
Someone must have tried.
But
well,
the
I guess you can't
tangles
do it
are
in people, but you could do it in a cell culture model. Can
So
you
now I'm
even
confused.
dephosphorylate the tangle?
These tangles now, thinking about how the tau is actually intracellular, most of it, unless we're wrong, but from what I read online and from here, it's intracellular. So the neurons themselves are tangled, or is it that the cytoplasm is huge? How is this tangle even forming if it's intracellular? Because the amyloid plaques, I understand.
So I think the amyloid plaque is almost working sort of in a prion-like way. And I am at least my imagination of what's going on with that tau tangle is microtubules polymerizing sort of inappropriately and, messing up cell transport and messing up just sort of a lot of intracellular motion because there's too much junk.
So then it's actual neurons that are tangled within themselves then, right? It's not, because an amyloid plaque is a physical formation of, it's a amyloid beta plaque. That's like an actual deposit
that stops signaling or stops transport in the neuron.
So Stanley Brusner has said that he thinks all these dementias are prion misfolding diseases. But they say here, while tau has been attributed prion-like properties, this behavior could be explained if the observed propagation results from tau following HSV protein distribution. So it could be HSV proteins are doing that, not the tau is self-misfolding, right? So I don't know. But this is, if tau is a key player, then you need to start studying it and how to reverse it or inhibit it, right? You know what the problem is? There's no mouse model. They're crappy mouse models for Alzheimer's where you overproduce variants of like tau, I think, and other proteins that give rise to
there are a few different genetic models, but none of them are great.
None of them are great, yeah. So anyway, it's cool. It's very interesting, right?
Yeah.
Yeah.
I wouldn't have guessed that.
So in my many tabs of Googling that I've been doing with this, I have an experiment plan. I have many experiments planned, but one of them I have no possible way of doing.
Getting a human brain?
No. Can somebody just do some viral infections in tau knockout mice?
Well, what would you look for?
Increased viral pathology, evidence that this immune response is actually
beneficial.
Useful.
Yeah, that's fine, but it doesn't tell you anything about Alzheimer's, right?
No, but it tells you if tau is an immune protein.
Yeah. Well, it sounds like here it protected neurons from dying. It sounds like it is, right?
Right.
One thing that I found interesting about the writing of this paper was that at the end of the introduction, the last paragraph that's sort of telling you what they're going to tell you, they reference all the figures. So they tell you the facts that they found and the figures, which I don't think I've seen that before. And then in the discussion section, they have a heading for every point that they're going to make in the discussion.
I actually liked that.
I did too. Like it was really interesting that that they did both of those things. It kind of makes up for talking about increases when they should have been talking about higher amounts. Well, and then another thing which we didn't mention, but it was when we got to some of those figures. I hate angled figure legends, and so, for example, figure 3C part 2. Oh my gosh, it was so confusing. And there would have been a way to label that figure without using angled figure legend things, and people just need to think about that more carefully. So,
should we do a couple of emails?
Sure.
I think that first one is too long, so
it's I was gonna say I wish not to read that. It's extremely long.
We can do the next 1, 2, 3, 4. 1, 2, 3. Yeah, we can do the next 4. So, Alan, could you start with Brian?
Sure. Brian writes, in TWiV 1179, what was that?
Go ahead. Sorry.
Okay. In TWiV 1179, Rich described how an AI program uses structure to detect unknown virus. Different DNA or amino acid sequences generate the same protein structures. Alan mentioned this is convergent evolution, but not always. Prokaryotes integrate sequences laterally. Evidence suggests that they use structure to preferentially integrate certain ones, probably as a way to repair damaged sequences. If the structure fits, use it. Structures with different sequences in prokaryotes may do similar things. This may not be convergent evolution. A clever bacterium is doing something more like eukaryotic sexual selection. Thanks for your great work, Brian. Very good point, Brian.
It's interesting. Using structure. Huh. I had no idea.
Yeah.
That's pretty cool. So when you have independent organisms, you get evolution of the same end protein. We call that
Convergent evolution.
Convergent evolution. But he's saying that Brian is saying that
prokaryotes, since they can transfer genes laterally between species, you've got this other mechanism that could be going on.
But all things can do that. Eukaryotes can transfer laterally too, right?
Not nearly as much.
Not as
much, but they do it.
It's been years since I exchanged genes with a plant.
Well, no, among say mammalian cells, they can do that. Yeah. And prokaryotes are, they're prokaryotes. Kathy, can you take the next one?
Lynn writes, Dear TWiVers, I was amused to hear you relate the Zinder story on a recent episode. I teach research ethics at UConn, and in the module on data sharing responsibilities, Peter Sutlow and I have used that story as a case study discussing the ethics of withholding research resources from the scientific community, as well as the ethics of obtaining such resources without appropriate permission. It's a perfect story to address both ends of that ethical spectrum. As a virologist whose career path took him into academic administration, I've thoroughly appreciated listening to TWiV and stirring my old virologist bones that had lain dormant for so long. You're doing important work and doing it well. Best wishes, Len.
So talking about sharing resources, I don't know if it used to be that Cell said, if you don't share a resource published in Cell, you can't publish in Cell anymore. They never enforced that. Never. And people would routinely not share their reagents. Anyway, I mean, I don't get it, Cell. Does anybody know what the state of that is now? Alan, you might know.
I don't know what the situation is with Cell. I do know that journal editors often have these policies, but they don't really have time or inclination to be the cops.
Yeah, what are you going to do?
And they certainly don't want to wade into some of the academic politics that gets involved in some of these. So I'm not sharing them with so-and-so, but I'll share them with so-and-so. So I think there's just kind of been a general approach historically to say, we will not tolerate this, and then look away.
Yeah, look away. So the phage story that I related is that someone wanted a phage from Norton Zinder, and he wouldn't give it to them. So they wrote him a letter.
Yes.
And requesting the phage, and he wrote back on a letter saying no, and the letter was contaminated with the phage, so that's how they got it. And,
I tell that story so often to my students.
And Len says that too is unethical. You shouldn't use stuff without permission. But I mean, shouldn't all resources be available to whatever scientist wants them?
I don't know that they used it without permission or sampled it from the environment.
The environment of the letter.
Right? I mean, you can take anything you receive or find and grind it up in the blender and see what's in it. Right?
Yeah. Maybe the only shortcoming was that they should have said, oh, by the way, we got this phage from your letter and we're going to use it.
Yeah, what are they going to do at that point?
Because they should have shared it in the first place.
Yeah.
I don't understand the not sharing of reagents and resources and like everything. It seems so, I don't know.
It's worry about getting scooped.
Yeah, people want to do it themselves.
It's like, just be better. I don't know. Just don't be worried about what other people are doing. Just do better.
If someone is doing it faster than you, that's life. Move on.
Exactly. Deal with it. Okay.
I gave stuff to people who ever wanted it.
And also, if you're a researcher, do you really have time to do all of the possible experiments with this reagent?
Some people are just driven, right?
Yeah.
They just don't want to do it. It's an unseemly part of science. Brianne, can you take the next one?
Sure. Sharon writes, Hello, TWiV team. I was intrigued to hear alpha-1 antitrypsin deficiency, A1-ATD, mentioned on TWiV. I'm a virologist and have some experience with A1-ATD from my work in an assay development lab at National Jewish Health in Denver, Colorado. NJH specifically sees patients with pulmonary, immunological, and cardiac diseases. Our lab focuses heavily on infectious diseases, especially mycobacteria and other lung pathogens, as well as other lung diseases like A1-ATD. Your listener Rachel wrote in about episode 1167, in which Dixon mentioned what Rachel and I presume is A1-ATD. I wanted to follow up on Rachel's great A1-ATD summary with more additional information related to A1ATD. A1ATD is quite common. 1 in 2,500 to 5,000 people of European descent have A1ATD. It's encoded by SERPIN-A1, which is a highly polymorphic gene. The degree of disease progression depends greatly on the SERPIN-A1 mutation. There are many pathogenic mutations, more than 40, and environmental factors. Not all mutations cause the protein to get trapped in the liver, but some mutations result in the protein not being made, null, or proteins released in the circulation with reduced inhibitory function. Mutations that confer A1ATD are the only known genetic causes of chronic obstructive pulmonary disease. A1ATD's main target is neutrophil elastase, NE. It inhibits NE activity mainly in the lung so that NE is not overactive and lung architecture is maintained. It is an acute phase protein that helps reduce NE activity post-infection. People with A1ATD are much more susceptible to pulmonary infections or pulmonary exacerbations from air pollutants, for example. I wasn't sure about Rachel's question regarding her pulmonary nephritis, but I found this paper and gives a link that suggests a link, especially in ZZ patients, severe A1ATD, which is probably why her physician mentioned a kidney biopsy. I also found this from another article and gives another article from Clinical Nephrology. Please pass this information along to Rachel. We have lots of great docs here at National Jewish Health that specialize on A1ATD if you ever consider doing an immune episode on it. Wishing you all the best in 2025. Thank you for keeping me up to date on the most recent virology advances. And Sharon is from National Jewish Health and the Advanced Diagnostic Laboratories there.
Cool.
Sounds great. And Angela, the last one.
Yeah, Molly writes, Dear TWiV team, I just wanted to write and say thank you for the work you do to help make science and specifically virology accessible to the general public to a general public audience, including colleagues working in different fields. I just finished a PhD on the politics of emerging infectious disease, surveillance, and response. While we have different politics, and I sometimes wish TWiV would cite experts from history, geography, and political science departments when discussing those topics, just as you insist people listen to scientists when speaking about science, the podcast was enormously helpful for making sure I was accurately describing infectious disease science while writing my dissertation in human geography. Your clearly explained debunkings of the various lab leak hypotheses of SARS-CoV-2 origins have been especially invaluable. I've recommended them to colleagues numerous times. On the left, I see a lot of people entertain versions of the lab leak theory because they have been harmed by medical practitioners or government institutions charged with protecting them, or because they study very long histories of violence carried out by these institutions against non-white people, disabled people, queer people, and other. For example, the 1927 Buck versus Bell decision legalizing the forced sterilization of disabled people in the United States has never been overturned. I mention this because I think for this segment of the population, it's not a matter of not understanding science, because the problem is that many folks have a well-founded and deep-seated mistrust of many government, medical, and public health authorities. So as much as I want to pull my hair out when I see, for example, respected award-winning investigative journalists entertaining versions of the lab leak theory, I also sort of get it. The way I've personally had luck reaching people who are coming from this perspective is to acknowledge, first and foremost, that their distrust makes sense. I then help them walk through some of the structural drivers of health inequities and failed public health responses relevant to COVID-19 so that they may understand how things like the lab leak theory actually distract from addressing those problems. And then I also, of course, direct them to relevant TWiV episodes so that they can better understand the science. For other academics, it's especially effective to tell them that one of the science papers establishing evidence for the market origin passed no fewer than 5 peer reviews. I don't know if people who haven't been through peer review appreciate just how difficult it is to satisfy 2 or 3 reviewers, let alone 5. Anyway, in sum, thanks again for all the work that you do. Best, Molly Holmberg, postdoctoral fellow from Balsillie School of International Affairs at Wilfrid Laurier University, which I think is here in Canada, or did I make that up?
I think it is.
I think so,
yes.
if you tell them there were 5 reviewers, they'll say, they had a conspiracy.
Well, no, they numbered the reviewers 1, 2, 3, and 3, and 3.
Exactly.
So reviewer 3 was intense.
Reviewer 3 was just like all over this.
So this point I want to address. While we have different politics, I sometimes wish TWiV would cite experts from history, geography, or political science departments when discussing these topics. I don't think we go into any of these topics in enough depth to do that in any way. That's not our strength, right? We do virology here. Mm-hmm.
And we can't cite them because we don't know of those experts. But I mean, maybe she means if we go into those topics, we should be educated with who said experts are, so we can then quote them.
Yes, right.
I think
the point she's making, and I do not have an exhaustive memory of everything ever said on TWiV, so it is entirely possible that at various points we have strayed a bit far into some of these fields where we maybe should have looked at what the experts in those say.
But I feel like most of the time we say, at least I think, or I'm not sure, but we're not saying things like as a matter of fact or that we know or trying to be experts in those fields. But either way, we will be careful.
Maybe she's just saying that when we might think about, or we might think that we're veering out of our lanes, to acknowledge that maybe this is something that we don't have all the expertise in. I don't know.
Yeah.
Mm-hmm. Which is fair. Definitely.
I really like her way of talking to people who are not sure about SARS-CoV-2 origins, and the ways that she links this into kind of structural inequality, and how she talks to people about that. Everything she describes here is lovely. I will probably steal some of this, Molly, and I highly am impressed and really want to give you a big thumbs up for coming up with this, because this is really good.
Good strategy.
And the university is a public university in Ontario with campus. The main campus, it sounds like, is in Waterloo, and the newer Brantford and Milton campuses.
Hey, let's do some picks of the week. Angela, what do you have for us?
So my pick is kind of like, I choose happiness. I know that sounds crazy. I choose the adult development, what is it called officially? Sorry, let me see. The adult development study from Harvard, which has been going on now for over 80 years. It started in 1938, I think, I recall seeing. Yes, 1938. And the current director at Harvard is Robert Waldinger. And there's actually a TED Talk by him, which is wonderful, on this study and on their main findings of this study. And it's also just a wonderful TED Talk. It's only like 12 minutes, and it's about just overall. So basically what they did is they started with, how many people? 74 men, I think. Yes, 74 men. And then those men then had over 2,000 children. And yes, there were only men in this study because at that time in 1938, that's how science
who you signed
was
up.
going. That's what you signed
That's
up.
who was at Harvard, right?
Yeah. It was at Harvard, and it was all men. So it was only men, and they followed them for 80 years. Some of them are still alive, or at least were when he gave the TED Talk, which would have been 10 years ago. They were in their 90s, and they had over 2,000 children from those original 74 men. And basically what they would do is they would get, from the moment of like Genesis, every 2 years, the experiment starts. Every 2 years, they would have medical exams, including CT exams, consults with their family, and then also with the actual person, so the man that was in the study, and blood work, etc. And they did this every 2 years for over 80 years. And in the end, they showed that good relationships keep people healthier and happier. And I know that this seems like common sense, but the fact that they have data to support it is very exciting, I think. And this is like an old study, but I came across it again when I was looking at some TED Talks, and I thought it would be nice. And that social connections are obviously increase survival, literally, in these people, and that loneliness kills. So maybe put down your phone, in the evenings and talk to someone instead of just looking at a screen. And some of the main predictors of health and old age was just happy meaningful relationships in middle age, actually. So those that didn't have happy, meaningful relationships in middle age died sooner. And I mean, it's just interesting. And it's actually one of the oldest studies that has ever been done. So like actual experiments. And it's still ongoing.
So if you're middle-aged and loner, you're screwed, is the philosophy.
I mean, just go talk to someone outside.
Sorry, no, you were trying to put a positive spin on this.
Go to the coffee shop and talk to someone, though. It's not even about only bonding, really, like having a partner or having children, but that it's also just quality of relationships with friends. So even just any sort of meaningful relationship.
So I was very curious about one thing that you said about the 2,000 children from the 72 men, because I did the math, and that was 27 children per man.
That's what he said on his TED Talk, but I think he means grandchildren.
So according to this website, it says that there were actually 724 people. Not 72.
Oh, sorry. Then I missed the two. I'm so
that makes a difference.
I'm so sorry.
Because I've been sitting here like 27.
It's true. It was in the hundreds. When I said 72, I wrote it in my notes and I was like, that doesn't seem like a lot. Well, okay, I'm sorry. There's a two.
Yeah, I was a little baffled. Did a little quick math.
Hundreds of people, which makes sense because that's like 3 or 4, or not even like 3-ish, 2 to 3, as opposed to 27. Okay, so hundreds of people were followed, and some of them are still alive. There's like very, very few that are still alive.
I feel I'm doing this through podcasting.
If it makes you happy, for sure.
I mean, all these podcast hosts, right? I talk to them regularly, and then I do the live stream, and lots of people come back every week. But then people visit here, and we have nice conversations, right? Randomly. So I don't know if I hadn't started, I wouldn't do that, right?
True.
Probably be a cranky retired academic. So
telling kids to get off your lawn.
Get off my lawn.
Literally.
Read the literature.
Read the literature.
Brianne, what do you have for us?
So I have something that someone recently sent me as a gift. Now, if I had been particularly on top of things, I would have actually remembered to bring it with me to my office. Not that good, though. But this is a little hair clip that looks like a beaker. And it's great. And it looks like a cute hair clip. I really enjoy wearing it around. And any person with longer hair. Yes, I see Angela's face.
Sorry, I just saw it and I was like, I want to buy this.
Right.
Click
on the forward arrow and then you see it in 3 dimensions better.
And there's a picture somewhere where it shows a person with it in their hair.
That's so cute.
If you scroll down a little bit, you can see photos from the reviewers.
I love having hair clips at the lab too, because you always have to like throw up your hair, and I'm always like, "Where's my elastic?" This is perfect.
It is the thing that you need if you have long hair in the lab.
So cute.
Yes, the one who's pictured works at a science museum, so she wears.
So they call it a beaker, but it's really an Erlenmeyer.
It is.
It's
really
true.
An
it is. I saw it is an.
That is true.
Yeah.
And they also say it is good for scientist, STEM, chemist, or physicist. I'm not sure how much physicists.
I don't really know, but I know that the person who sent it to me as a gift was clearly spot on because it is exactly what I need.
Yes.
That's so
Brianne, they
really
make
cool.
an antibody hair clip too.
Well,
they do?
Okay.
Yeah,
my hair is going to be so well
I just clicked on one of the ads at the bottom, and it's bispecific antibody because it's a blue and a red.
Oh, look
arm.
at that!
Do you find it? Yeah.
Yeah, and there's a chromosome too.
Mm-hmm.
And there's
cool.
a syringe. Okay, there's a whole bunch of them. We're gonna have to look through this.
There's even a gel, an SDS
There's a polyacrylamide gel.
Oh my gosh! What does that look like? Just square?
It's a rectangle. It's got the loading lanes across the top and some blue. It's nice.
There's also
cool.
a grilled cheese hair clip.
There's some pictures of people wearing them. That's great. One of them is looks like a blood bag.
I see that.
That's a little.
That's very funny.
Yeah.
It's cool. Bispecific antibodies. Nice. Kathy, what do you have for us?
I picked something that's related to something I know I've picked before. And so David Zinn is our local street artist, chalk artist. And I think, Vincent, you follow him on Instagram.
Yep.
And because Rich talked recently about pareidolia, it reminded me that David Zinn talks about it in his TEDx talk, which is only about 20 minutes long, but it's very entertaining, and he really does a good job of explaining what pareidolia is. And then I also linked to his page because you could check out some of his items. And pareidolia, if you missed that TWiV that Rich talked about it, is the tendency to perceive a specific, often a meaningful image in a random or ambiguous visual pattern. So looking at clouds and seeing something specific. And David talks about how he got into doing his street chalk art because of seeing things in his different way.
They're so
So
cute.
cool.
The cover of the 2025 calendar of the little mouse looking out of the brick is so adorable. Oh my gosh.
Yes. I bought 10 of those every year I buy more from David, and then because my brother knew him first and I kind of know him by association, but he always writes some little thing and then does a little sketch scribble, so I'm saving those. Kind of like autographs from David.
That's cool. Alan, what do you have for us?
I have a news article about something that I have tracked over the years. This is about screwworm, Cochleomya hominivorax. And this is it's a fly, it's not a worm. So it's a fly that causes a furuncular myiasis, which is a horrifying condition. The fly lays its eggs in any kind of open wound, even something as small as an insect bite on some mammal, and often it's some large herbivore because they're easy targets. Once the eggs are in, they hatch and they're nice and warm, and then they eat their way out of the flesh of the animal through the skin and reemerge and repeat the cycle.
It's like worse than a botfly. Somehow it's worse than a botfly.
This is absolutely horrifying, and it used to be very common in cattle all over the place. In the 1950s, it was eradicated from most of the Americas all the way down to Panama. And this was actually work done that was supervised by my grandfather. One of his people was a fellow named Ed Nippling, who came up with this incredibly clever idea, completely original at the time. We now talk about using this in gene drives, called the sterile insect technique. It turns out that this fly, the females only mate once, and then they go lay their eggs. So if you release a bunch of sterile males, they will mate with the females, who then go lay eggs that will never hatch. And you do this enough cycles, and you drive the fly to extinction. And this was done, it was a huge, huge program to breed up these flies and render them sterile, which is not an easy thing to do, and then release them. And this was done successively over a period of years. We wiped it out all the way down to the Darien Gap in Panama. And for decades, we've continued to maintain, the U.S. has continued to fund a program in Panama that continues releasing sterile male Cochleomya to keep it from coming north. Unfortunately, even though that program is still going on, the fly seems to have escaped. It has now migrated its way north and is almost up to Mexico. And this is a very good article in Wired that explains how that apparently happened. It seems that there's an active and growing business in cattle smuggling. And this happens across international borders, and a lot of these cattle are coming from further south in South America, where this fly is still found. And so they got smuggled north and then spread it, and once it passed the barrier in Panama, it was able to continue north. So not exactly an upbeat story, but something that I think is interesting and definitely worth keeping track of.
Yeah, I remember you saying a long time ago, was it your uncle My
grandfather.
Your grandfather worked on
Walter
this.
Dove. He was a medical entomologist back when that term barely existed. But this was a project that happened under him. He had some other stuff that he did that people have probably heard of too. But yeah, this was, until it was wiped out in the 50s, this was a huge economic strain on ranchers and dairy farmers. Because when this happens, obviously it does a lot of damage to the cow, and you can get secondary infections. And Angela, you've undoubtedly learned about this. So it's it's a bad scene, and then that causes a lot of knock-on effects.
I'm sure Dixon would also know
Dixon
for sure about this, and he would also have more to contribite, at least about like the, its distribution and yeah.
It's unfortunate. That's what moving animals illegally will do.
Yep.
My pick is a article in Science-Based Medicine by Jonathan Howard. It's called Dr. Jay Bhattacharya was nice to someone during a debate. That doesn't erase the consequences of his misinformation. So I just want you to read this and learn more about Bhattacharya, because people are saying, oh, he's a Stanford professor, he should be fined as NIH head. No, he's done too many ridiculous things. So, for example, there's this organization called Biosafety Now. Members of this organization care deeply about where SARS-CoV-2 came from, but are utterly indifferent to how the virus was handled after that. And they have partnered with Marty Makary, who's been nominated for FDA head, and Jay Bhattacharya, who have zero expertise in biosafety, but instead spread copious COVID misinformation and advocated for herd immunity via mass infection. So there's all these tidbits. For example, Bhattacharya wasn't transparent that the Great Barrington Declaration, which we covered on TWiV, was sponsored by a proud child labor advocate. And rather than admit his early pandemic pronouncements were erroneous, he tried to obscure them by claiming words have no meaning. And all he ever cared about was poor children. He was opaque and untrustworthy throughout the pandemic, and there's no reason to think he will change once he gets to the NIH. Anyway, you read it and understand why he should not be NIH director. But I'm not sure we can reject all these people, right?
That's so worrisome.
RFK Jr. is probably worse.
True.
We have a listener.
You guys should just come to Canada. Maybe you should just come this way.
Yeah.
I don't know if it's that much better. I mean, we have our own problems, but
Kathy, you want to do the listener pick?
Sure. So our friend Sarah wrote about this. It's a citizen science thing, and it's the Extremophile campaign. In your home, citizen science. And so we've put a link in here. The first thing you do is you sign up with Citizen Science. And if you don't like the one I'm going to tell you about, evidently you can sign up for lots of other citizen science things. But the gist of it is that once you sign up for the Extremophiles project in particular, you would be prompted to take photos of slime, crusty mats, stringy growth in your house, particularly in places where there are extreme hot-cold cycles, like washing machines, refrigerator drip lines, hot water heaters, solar panels, shower heads, maybe even your dishwasher. And then if the researchers, then you send those pictures back, and if the researchers see something promisingly unusual, they'll mail you with a testing kit, which looks to me like it's got some 50 mil conical tubes in it, and gloves and a scraper, and a prepaid envelope to send it back. And then they're going to sequence the DNA and add it to an open source data of microbes and catalog the diversity of what they find, and that maybe some might be useful for human endeavors. And the main one that I think attracted us in the first place is that it might be able to identify something that would tackle greenhouse gas emissions or clean up pollution. And they make the comment partway through the article that they might have found the Thermus aquaticus bacteria in home hot water heaters if people had looked, instead of having to go to Yellowstone. And that Thermus aquaticus, of course, is the bacteria from which we get Taq polymerase. So it's just a cool thing. You take some pictures, send them in, and maybe they'll sample something and they'll find some interesting extremophiles in your house.
Cool.
I don't know how I feel about sending them pictures of slime.
Pictures of dirty things in your house.
Yeah, it's like I want to clean it if I see it.
But I'm actually wondering, those times when you get that really weird like contamination in something in the lab that looks gross. Oh, yeah. I wonder if they would like a picture of that.
Yeah, maybe.
Like when you open the fridge and there's a beaker that's like has this floating ball of who knows what.
Yeah, exactly.
Like one of this, and you're like, I'm never opening that.
How many times my so-called bottle of sterile water on my bench would have something growing in it?
Yes.
It's like, how does this happen?
Yeah.
We had something when I was in the lab, Vincent. We actually had something, a little ball-like thing, start to grow in the still, in the water distillation tank.
Oh wow! Well, that's unfortunate.
How?
Yeah.
We just cleaned it out.
Yeah.
Shouldn't. All right, that's TWiV 1187. You can find show notes at microbe.tv/TWiV. You can send questions, comments, picks of the week to twiv@microbe.tv. And if you enjoy this and all our other science programs, we'd love to have your support. Microbe.tv/contribute. Brianne Barker is at Drew University, BioProf Barker on Blue Sky. Thanks, Brianne.
Thanks. I learned a lot.
Kathy Spindler is Professor Emerita at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thank you. This was a lot of fun.
Angela Mingarelli is at McGill University. Immune Vet. Is that Twitter or Blue Sky? Immune Vet.
It's X. Yes, it's Twitter.
X. I'm not calling it X.
Okay, well, Twitter. Okay,
the hell with
call it Zitter.
Thank you, Angela.
Thank you. I had a great time.
And Alan Dove is at alandove.com, turbidblack.com. Thanks, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV. Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.